Q&A panel, not the chat window. Be sure to include the speaker's name at the beginning of your question so we know to whom the question is being directed. Again, once you open the Q&A panel, click to type in your question. And we do have time scheduled at the end of each presentation to do some live Q&A with the speaker and our moderator. And then after the presentations, the presenters will do their best to type their answers to your questions. If they do type an answer to your question, you can see that in the My Questions column of your Q&A panel. So here is an overview of our agenda for the day. We will have some brief introductory remarks followed by 2 talks. We'll have a brief break from 9:55 to 10:05 a.m. Pacific time, then 1 more talk before our panel discussion. We do hope that you can stay for the entire program, but if you have to step away, we will be posting a full recording of the event on the event website and on our YouTube channel after the event. Okay, and as we head to the top of the hour, I am very happy to introduce our moderator for the first part of our program today, Dr. Kathy Richmond, Executive Vice President and Director of the Paul G. Allen Frontiers Group and Office of Science and Technology at the Allen Institute. Please feel free to share welcomes for Kathy in the chat. Welcome, Kathy.
Thank you, Megan, and welcome everyone to Exploring Frontiers, Predicting Biology, Translating from Mouse to Human Models. As Megan noted, my name is Kathy Richmond. I'm Executive Vice President and leader of the Paul J. Allen Frontiers Group, a division of the Allen Institute. The Frontiers Group upholds our founders' commitment to tackling the toughest questions in bioscience beyond the Institute walls by directing funding to investigators and centers from across the country and internationally. Now, we're delighted to have you virtually today for this symposium. Today's session will be spotlighting pioneering approaches to improve our ability to make inferences between mouse and human models, an area where advances could not only move the needle in translational efforts, but also in our understanding of fundamental biology. Now, normally we'd be hosting this event in person at the Allen Institute, but since we're virtual today, let me tell you just a little bit more about us. In 2003, the Allen Institute for Brain Science, launched by our visionary founder, Paul G. Allen, was built on the core principles of big science, team science, and open science. And since then, it's become known worldwide for open resources, including gene expression maps of mouse and human brains. Now, keeping to these core principles, in 2014, the Allen Institute expanded to include the Allen Institute for Cell Science, and that was followed in 2018 with the launch of the Allen Institute for Immunology. And combined, we became the Allen Institute. These 3 divisions all operate in our Seattle headquarters and take on large-scale foundational questions in bioscience, tackling them with multidisciplinary teams, and sharing that data and resources generated openly with the wider community. Now, working side by side with those efforts, our focus at the Frontiers Group is always about unearthing ideas that have the potential to open new fields and anticipating that next great breakthrough. So we are very excited about the program today. Now, model systems have been used successfully to pioneer foundational research, leading not only to advances in understanding of biological systems, but also to those really important translational breakthroughs. However, only a portion of scientific results seen in mice models are mirrored in humans. And as often pointed out, mice are not small humans. So how can we better infer which observations will hold true in both systems? Well, today's speakers will address just that. And I invite you to be part of the conversation. If something resonates with you, call that out in the chat. If you have a question for our speakers, plug it into the Q&A as Megan instructed. And at the end, we will have a panel discussion with the speakers moderated by Dr. Stacey Finley, and it bodes to be a lively exchange. Now, before we start our program, we'd like to especially thank our late founder, Paul G. Allen, without whom this institute, this event, and much of our research wouldn't be possible. So without further ado, I'll introduce our first speaker. Today, we will be leading off with Shai Shen-Orr, an associate professor at the Faculty of Medicine at Technion, Israel Institute of Technology, where he heads the Systems, Immunology, and Precision Medicine Laboratory since 2012. His research is focused on charting the immune system landscape namely the principles by which the immune system varies over time as a function of environment and genetics. For this, his lab develops novel computational methodologies, empowering human immune monitoring for precision medicine. Dr. Shen-Orr's presentation is titled Found in Translation, Leveraging Machine Learning Models to Bridge the Cross Species. And it's my honor to welcome Shai. Take it away. There you
hear me now? So a real pleasure to be at this symposium today. It's a favorite topic of mine. Bridging the cross-species gap is basically, I think, one of the big challenges for computational biology today, and there's a huge amount of opportunities here. Before I get going, I'll just say I'm a faculty member of Detection, but I'm also a disclaimer. I'm the chief scientist of Cytoreason, company that's kind of been commercializing the computational solutions that we've been developing in the lab for drug development. And you know, Kathy basically already said, in many ways, this symposium is all about how we have this gap, the cross-species gap, the ability that we need to be mindful of every piece of research that we do. There's many different confounders that lead to this, and I've listed them here in two divides. On the left-hand side, you see those kind of traditional imposed gaps that really come from the way we've traditionally kind of done science, such as mice that are have grown in a laboratory condition versus a real-world exposure in humans. For our convenience, we've kind of developed, we've limited the diversity up until recently. We have developed work practices that really put the mouse at the forefront of our research, and it's difficult for us as humans, as individuals, to change our practices because of past commitments, because of what our skills are as the landscape of research changes. And in the end, there's a difficulty to actually understand the limits of what is similar versus not in mice and how they can be used. So in many ways, the traditional gaps are being addressed, and I imagine that Nadia today in her talk will talk some of the great work that's being done in the Jackson Labs and others to try and make mice more kind of better models to model human diseases. But there's the difficulty that sits on the other side, and that difficulty is something that's does not have we cannot change just based on changing our traditions, and those are the biological gaps. So there's basically differences in lifestyle. These are our two species have evolved in very different conditions. If it's just about the lifespan or the height that you're off the ground, just that is going to affect, from an evolutionary perspective, what's going to be the constraints on the species. And we're talking about two species that are on the order of about 100 million years apart. When we're looking at disease models, we suffer from the problem that we may see a model that has a similar phenotype, but not necessarily molecular similarity. And or if we do devise genetic models, they are very partial in that they are usually have only one or a few genes that have been made similar to what we see in humans versus the entire network that may actually be different. And so it's the biological gaps are in many ways, I think, when we think about where we can kind of infer better, at least from a computational perspective, that those are where some of that information can come in, because there's no way to overcome them really outside of that. So it's kind of loading slowly here, but the screen is not updating on the. Oops. Okay, so so the utility of mouse models is a much debated issue, and what I illustrated in the slides are two papers that actually were published in PNAS a few years back, really, if I remember correctly, about a week apart. And if you can see the titles here on the left-hand side, The title was Conservation and Divergence of Transcriptional Programs of Humans and Mouse Immune Systems. We in the lab used to call this paper Mouse Rocks. Basically, it says, look, mouse and humans are really conserved. Only 169 genes, if I remember correctly, are reported to be significantly different when you look in particular kind of across different cell types. On the other hand, on the right-hand side is a paper that made it all the way to New York Times, where basically the paper reported that mice are poor models for human disease. And we call this the mouse sucks paper. And in many ways, when you look at these two papers, really they're really going head to head, but they're not arguing about the same thing because the utility of looking at conservation of cells or looking at disease similarity is not really the same. So if we think about utility and if we think about models, George Box said a statement really for statistics, but implies for animal models as well. All models are wrong, but some are useful. And so we need to be thinking about the utility of this. And this becomes difficult when we realize that this is a changing landscape. So if you think about historically, human data was never available, and the breadth of legitimate animal model use was very big, because if you wanted to do research that would be kind of biomedically relevant for discovery and in the end therapeutics for human, there was no way to do it really in humans. Over time, and especially now, it's become very easy to generate data in humans compared to what it used to be. And with that, the utility of animal models, of where they get used and how we should be using them, changes. And it's difficult for us to think about this as we are creatures of habit, and we get going with kind of a particular way of how we do our research, and yet the landscape changes and we need to keep up. So the utility has been changing, and just to kind of give an example of this, if you think about drug development and the stages from preclinical to clinical stages, there's in the preclinical kind of the non-human stage, there's uses of animal models for discovery, and there's uses of animal models for safety and efficacy post actually deciding on the indication, deciding on the target, deriving a molecule, and so forth. And just as an example of something that's changed, and we've been working on the commercial end, is the ability to actually have human-generated data has really transformed so that we can take all the human data, take all the data that's collected in clinical trials in the public domain and so forth, and really affect how we, the early stages of discovery. Why discover in an animal model where you can first browse computational models of human diseases? On the other hand, there are places where we're not even going to get, there's no way to kind of get around the fact that we want to be checking safety and efficacy of drugs before we put them into humans. And those are early stages that are not going to go away anytime soon. So differences in utility will be important, and how we think about this as a greater picture is ever changing. Now the point here is that if we think about the complexity of what I just described to you, all these different gaps and so forth that exist, and the availability of data, the concern that we have in mind is that in the end, these species are different from a biological perspective, and we have a very particular utility. It doesn't matter right now if it's for drug development or for other reasons. The point is that in the end, when I need to kind of look at a mouse model and I want to infer something on a human, the initial conditions are different. Again, doesn't matter why the gap exists. And I want to get it right. And the differences, even small differences, are going to affect in the end what I would predict would be relevant. So the question is, can you overcome the fact that in the end, even if I mutated the exact same gene, the network of mice and humans is very different, the conditions are different, and so forth. And in many ways, the fact that we've been displacing mice in some places by generating human data is also the opportunity to do this well. And so what we've been developing in the lab, and this is work first initiated by a PhD student in the lab of Shai Norman, now a postdoc at the Broad. Not sure if it's updating on the screen. It's taking a bit. So this is a model that we call found in translation. And found in translation takes the idea that if I'm generating a mouse experiment, if I'm doing work in a mouse and doing some experiment in a mouse, ultimately what I want to do in the end is understand the human disease, and rather than doing this traditionally, but just by looking at the mouse result and trying to infer in human, we can leverage the avalanche of data that exists now on human diseases and their mouse model equivalents. And we call this found in translation, basically using a big prior of all of existing knowledge to try and infer better from my new mouse experiment that I did in the lab because I had to do it for particular utility to what would happen in the human. And so there, just to kind of show you the work we've been doing this, I'll kind of describe this in the next few slides. What we did is we basically built a large database of human measurements of gene expression measured by both microarrays and RNA-seq, all available in the public domain, human experiments of disease and healthy controls, and the equivalent mouse model of that disease. So people are trying to study the disease and they're using a particular mouse model to do it. Again, the experiments would have measurements of all the gene expression in a particular disease and controls. And we paired mouse models with their human disease counterparts so that we had 170 cross-species pairs. We call them CSPs. And we look at what are, for the 20,000 genes that are being measured, we are trying to compute what their effect size is and what genes are really kind of changing between disease and healthy, doing this across 28 different diseases, high quality data sets, doing a lot of manual processing of making sure that we got good data, and we carefully are pairing the cross-species the cross-species experiment. And I'll say here, note that the utility is the acceptance that it doesn't matter if a mouse model is good or bad. There's no such thing as a mouse rocks or a mouse sucks paper. The only thing is that the researcher did a mouse experiment, and can we somehow help that researcher do better work by leveraging the computational model of all the prior disease. And so this is a training set, and really what you now can do is a lot of different comparisons across many different cross-species pairs, across many different false discovery rates, statistical significance, and comparing fold changes in Q value or the equivalent of a P value, and inferring in every given comparison what is the human differentially expressed genes, we call them DEGs, or the mouse differentially expressed genes. Here I'll show the laser pointer here, and that this overlap we call basically the true positives, where the mouse model represents the human disease. In terms of overlap, it's easy for us to to infer for those genes. And so when you do this comparison across many different diseases and pairs, you can start analyzing all the data to kind of get a sense of where this is taking you. Long story short, across many different fold changes in Q values, the true positive fraction, where the mouse genes are telling you what's going to happen in the human disease 1-to-1, the genes in the mouse go up in the disease, and the genes in the human disease do the same. You're talking about, on average, about 1 in 20 differentially expressed genes are shared between the two different species. And I'm not going to argue if this is good or bad. It doesn't matter. The question is, can I somehow do better than this? Can I use data to infer better what would happen in the human? So shifting here. So we can now look at the kind of a distribution of how genes behave in the mouse, the effect size of disease versus control, and in the human. And we can see genes that are relatively well correlated, not great correlation, but you're bridging the cross-species gap. And you can see genes that behave opposite. And this happens at the gene level, and it happens at the pathway level. So basically, even if you take an aggregate of genes and try to infer what's going to happen, it's not necessarily the case that you will succeed. Often you see no correlation or negative correlation and so forth. And you can see this on the distribution here on the examples as well. But again, that's the state of what we can infer from the gene expression data. And the question is, can we do better? And so to do so, we kind of devised this idea of the found in translation algorithm. How does it work? Very briefly, you give me a mouse experiment that you did in the lab because you decided that this is the right experiment to do. And I'm going to take this large database of all human disease and mouse models that exist in the public domain. And I'm going to take your mouse experiment. I'm going to re-rank your genes based on all the prior of everything that we know about this, about how these genes behave. We've even added in a classifier that allows me to do whether I can do this this procedure in a way that's going to be beneficial for you, for your mouse experiment. And then I'm going to be, the fit model is going to predict what are going to be the genes that are relevant in human. And so that basically at zero cost, we can, just running a computer program, we can generate a better prediction or a prediction of how your mouse model is going to behave when we think about the translation to human. Again, just to kind of to explain this in a more technical manner, think about taking a database of all the kind of mouse experiments, the mouse effect size. You can see it here on the x-axis, and the human disease versus control counterpart. You can see it here on the y-axis. And we just resample from this distribution all the time, trying to learn what is the relationship between how the gene behaves in the mouse. If it goes up, what's going to happen in the human? Is the gene going to go up in the human or not? Is it going to go down and so forth? I just want to generate statistics on this. I want to understand how the gene behaves. And once we've done that, we can now, if you give me a new mouse experiment, for every gene, I can try and take the new mouse result that you derived in your experiment and overlay this on the axis to generate a distribution of predictions for which there is a mean that allows me to define what is the prediction and what's the confidence interval for this prediction. So this is the idea of fit, and kind of show you how this performs. So just taking a lot of different genes in the genome and asking what's the mean standard error of trying to predict what's going to happen across all different genes. In red, you can see what happens when I just take the straight mouse results and try to go to human versus what happens when I use fit. Basically, the error drops. And if this goes down to a single gene level, and just to show you how one gene behaves, you can take a gene like SDF2 in a disease such as sepsis. You take all the database of everything you've learned, leave all sepsis data sets aside so that you're sure that you're not contaminating your data with some knowledge that may influence your prediction capability. You look at how the gene behaves across all the different data sets. You can basically see that SDF2 has a negative correlation between mouse and humans. When it goes up in mouse, it goes down in human in the reverse. We can overlay here on the middle what the SDF2 genes look like in sepsis, but we're not putting them into the model. These are every red point here is one sepsis experiment, kind of one cross-species pair where we have a mouse model for sepsis and a human sepsis data set, and we're trying to predict, and we just kind of overlay to see what happens. And now, given the new mouse experiment and this model of how the gene behaves, we predict the kind of where the gene would fall. And basically, in this case, you can see we can predict a negative relationship just based on this prior data. So this is what fit looks like at a single gene level. And if I do this for many different genes, you can see the value of using prior data in this manner. On the left-hand side is a single kind of sepsis experiment looking at the mouse fold change on the x-axis and the human cross-species pair counterpart. Again, the data is left out. The model does not see this when it makes the prediction. And you can see the overlapping genes in black between mouse and humans. These are the true positives, where the mouse successfully helped you understand what's going on in human. Now you apply fit gene per gene for this type of behavior, and you can rescue all the yellow genes that you thought would not be relevant. And turns out that by using this prior data, you can identify them and rescue their signal. So this is the basic idea. Again, just moving this across many different thresholds to try to compute. You can see what the overlap, in this case in IBD, between a mouse experiment and the human in the blue line. And if I apply fit, I can improve this, and that's the kind of light red line. And if I use the fact that I know what my confidence interval is, and I just take the top, say 30% of the genes that have this best confidence interval, I can more than triple my success rate in predicting what genes are going to be relevant for the human. And so taking this across all the different diseases in our data set, leaving out each disease every one disease at a time so that we're sure we're not contaminating it, we can generate the ability of our computational machine learning model to predict how well what's going to happen in the human. In cyan, you can see what happens when I just compare two, when I just use the mouse straight on. In black is what happens when you compare a human data set to another human data set. So this is, you can see how much noise exists in biology by comparing two human data sets of the same disease. But in pink and in red is what happens when you use all this information about the prior models that allows you to boost your signal by up to 20 to 50%, and really kind of discover new biology that you would have missed in your mouse experiment. Now, this is great news. We got really excited, but turns out that it doesn't always work. It works better in some experimental conditions in some disease models than others. You can see on the left-hand side, mostly in inflammatory conditions, better than cancer and some kind of diseases that are such as autism and others. And we tried to figure out, well, this is a problem. Obviously, what really affected it, we realized, is the size of our training set. There's a bigger resource that needs to be built to do this well. But the important thing is, can I know ahead of time whether the model, whether fit is going to be of use for my mouse model, the one that I generated in the lab? And so we built a classifier that allows us to predict fit's performance before applying it, so that I want to know if I should or should not use my computational model to learn. Maybe my mouse model cannot be, would actually be hurt by leveraging all the prior data. And so doing this, we basically were able to derive a classifier that predicts with 80% accuracy whether applying the fit model is going to be of use for the researcher or not. And so this really allows us to use this in an intelligent manner where it's going to be of impact in making discoveries. So just to kind of show you what this looks like, shown here are two plots from what's called a gene set enrichment analysis. For those of you not familiar, this is a computation that's done to try to discover which pathways or sets of genes or kind of new biology exists in your data, when measuring, say, the entire genome by mRNA. And you can see the mouse results of taking a mouse experiment, straight mouse experiment, analyzing it and trying to see, can I discover biology? And the blue line showing you what happens when before taking instead of applying the mouse results directly, you feed them into the fit model and then try and recompute, zero cost, just recompute what the biology is. And you can see here, for instance, we can identify that complement would be significantly upregulated or antigen processing in this experiment. And you would miss this just by taking the mouse results out. To really kind of make sure that this is bringing value, we decided to go for something risky and orthogonal. And instead of looking at kind of a set of genes, we said, well, could we do an experiment using a complementary approach that would validate a fit prediction? And to do so, we wanted to make sure that nobody thought that we were influenced by the human disease. And so we looked for genes that the mouse result was not significant. Applying fit would tell you that this gene is involved in the disease, and we had no human data that would suggest that the disease that the gene is upregulated in the human. So basically, either it's we are wrong and the gene isn't upregulated, fit is wrong and the gene isn't upregulated in human, or maybe the human data wasn't representative as we'd like. And so we found a bunch of genes like this. So recognizing human may be basically a silver standard, we predicted a variety of different genes and decided to validate on ILF3. ILF3 is a gene that's actually been associated with RA and psoriasis, but never with inflammatory bowel disease. Our fit model of applying mouse IBD data that did not have ILF3 is differentially expressed. Applying this into using fit would suggest that ILF3 is differentially expressed. And so we bought an expensive antibody that I had no use for insides from checking that our model works. And we stained healthy and IBD patients, and you can see the protein predictions on the right. So from mRNA in mouse to human protein, suggesting that IL-3 is indeed playing a role in IBD, in human IBD. I'll just wrap up by saying that this really is an open source package. You can apply it either through a website, mouse2man.org, or through GitHub. We think this is just the first step in this. There's way more that can be done. And in fact, I will say with a kind of a take-home message that the tides are changing and we need to really figure out the use cases. There are many different use cases for mouse, and each of them can be tailored with a computational approach that would fit it. This computational approaches really come at zero experimental cost, and we need to recognize that the human data may be silver standard. So there's certainly the benefit in the mouse. And we present here an idea of how you can basically reuse data through building these resources that let us leverage and can be extended, of course, to networks, a single cell, to incorporating other priors. You'll hear some of that later today, or modeling different conditions. And really, I think for the people in the audience who are in the field of computational biology, I have to say that one of the things that excites me the most is I think this is a new frontier for computational biology. It's a place that now that the data exists, there's a huge amount to do, and there's a huge need for across the entire field of biomedical research, not just in mice, but also in other animal models. So with this, I'll just thank you for listening in. The people that appear here in gold also played a role. The first author, as I said, is Heli Normand. Happy to take questions if there's time.
Thank you so much, Shai. We have time perhaps for one question. There's a stack of questions in the Q&A, but the one that's been upvoted from Martin Borg Jensen is, it sounds like FIT only considers translation of individual genes independently. Do you have thoughts on modeling translation of biological activity at the levels of groups of genes? The translation might be more robust at that level, with different related genes being more or less important per species.
Right. So it's a wonderful question, Martin. Certainly we have. FIT was basically the first application of doing this type of thing, and we wanted to keep it simple. It's not a trivial task. We have been, in fact, playing with doing this for groups of genes. In often cases, it does perform better. Not always, and that's one of the reasons we're investigating this. We think we need to kind of put in not just the groups, but really kind of modeling the networks in many ways, and sometimes this can really drive your signal.
Thank you. Well, there's more questions that Shai will hopefully be able to answer while we move on to our next presentation. And our next speaker I'm excited to introduce is Nadia Rosenthal, the Scientific Director of the Jackson Laboratory in Bar Harbor, Maine, who is a global leader in the use of targeted mutagenesis in mice to investigate muscle development, disease, and repair. Her research team has recently shown how modifying the profile of immune cell infiltration can facilitate or prevent tissue repair, and they have demonstrated the power of harnessing mouse genetic diversity to identify new genetic pathways in the varied response of cardiovascular disease. So Dr. Rosenthal's presentation is titled "The Mouse Ascending, Translating Human Genetic Variation to Function." Welcome, Nadia.
Thank you so much. Thanks so much to the organizers for including me in this really exciting event. I'm actually much more interested in hearing the other two speakers than telling you my story, but I'm going to tell you my story because I think there's some interesting issues associated with the translation from mouse to human that I can perhaps elucidate and follow on Shai's fantastic story. So the issue of human variation is really no more clearly evident than in our response to disease. So the big challenge we have is that we're very, very complex, and no one in the world is exactly like you. We differ at every level of our biology, from the our appearance right down to our organs, our tissues, and the genes that affect us. We saw that in some of Shai's data. And of course, the problem is that disease then can have various ways of manifesting itself. So if we were able to really project your individual health roadmap, we'd need all the mutations in your DNA. We'd need all of the variants in your genetic makeup. We'd need your entire lifetime exposure to environmental stimuli and challenges. We'd need to know about every organ in your body and how it responds to diseases. And ultimately, we'd also need to know how that is all affected by aging. So can you imagine that with all that data, we could move very rapidly through the kind of translation that Shai was telling us about? The problem is that as humans, we really are okay with our mutation knowledge. It's getting better and better with GWAS. Not so great with diversity, although we're getting better at that. The real problem is when we get down to environment, we're so different in terms of our environmental stimuli that it gets more complicated. And of course, we're not going to take out people's brains and hearts to see how their tissues respond the way we do with animal models. And aging is also a problem. So ultimately, what this means is that we really need other solutions to getting this information. And so for many, many years, we've been using mice, as we heard from Shai. The inbred mice are extraordinarily useful. They're homozygous. They're sort of like little clones. They're genetically identical. They're reproducible in models, good, bad, or not. The problem is that each one of them has its own sort of dark story. I mean, for instance, here, just a few mice that you might have used in your laboratory, AJ, they actually have a lot of problems because they're inbred. So in this case, if you're a cancer biologist, you really have trouble because you've got this background problem of lung adenoma, or a C3H mouse, which has spontaneous hepatomas. And then, of course, there's the famous C57 black 6J, which, of course, we at Jackson Laboratory love to sell you. But the point is that if you look at some of the problems with this mice, it's not exactly your normal guy next door. You know, it has a low pain threshold, it's alcoholic, it's neurotic, it's diabetic, it loses its sight, its hearing, and low bone density. So many of these are very relevant for humans, but they're not relevant for every human. And this is the major problem we have. So why stop with one mouse? Why not look at many different mice? And so my story today is going to be about harnessing genetic diversity in the mouse kingdom for understanding better how we can translate information that we see in the human to the mouse, as well as the other way around. So the concept here is that if you're studying a lot of humans, why would you want to study just one kind of mouse, one strain? It's like taking any one of us in the audience and saying that that's going to be the gold standard for everybody else in the world. So indeed, it's not very hard to imagine how a number of different genetically distinct mice could better model a human population, and in particular, model the diseases they get. Just to very quickly introduce one of the most sort of long-standing examples of this, we have a group of mice that are actually inbred from 8 different strains of mice that were initially inbred, obviously, in many labs. And we then, as a consortium, and I say the royal we here because I wasn't involved in the instigation of this, it really started 15 years ago, taking 8 different strains of mice and mating them together to mix up their chromosomes. And ultimately, after about 20 generations of this, then inbreeding them by back crossing in order to make distinct recombinant inbred strains. So this has the advantage of clonality. It means that the animals are indeed inbred, which has some problems, but it also allows us to relate each inbred strain to another inbred strain. It's a bit the way in which human population geneticists have exploited some of the same kind of allelic restrictions in places like Finland or Iceland. So these recombinant inbred strains have been very useful in a number of cases, and I'm going to use some unpublished, or I should say, published on bioRxiv only, not yet rejected from our favorite journal, to tell you a little bit about an example in which infectious disease has been very, very effectively modeled using this approach. And here's just an example of a huge number of papers that have come out in the last, let's say, decade that really do recapitulate the variation that we see in human infectious disease outcomes. And you can see here influenza, Mycobacterium, tuberculosis, SARS, and others, West Nile, et cetera, Ebola. So each one of these examples shows how if you survey a number of different genetically distinct mice, you get a much more realistic look at the pathways involved. Where are they similar? Where are they different? And of course, that can then hopefully translate back to human. So, I mean, this is really a very young field. So what I'm telling you more is potential rather than all the beautiful analysis that you saw from Shai. But I think that maybe these animals could actually enrich some of what he's doing as well. So I'm going to focus in on COVID-19 because we've just finished a study looking at the actual effects of SARS-CoV-2 infection on genetically distinct mouse populations. And I don't have to tell you what the problems are in COVID-19. We have huge variation in the way that people respond, affected by age, sex, environmental exposure, all of those variations I talked about before, and most importantly, by genetic makeup. That's been hard to grasp, although it's getting better. But in the long run, we really do need to know what are the genetic components of the response to this virus, because that would allow us to do much more predictive work. And we know that host response defines the disease severity. That's something that is really clinically obvious. So the big question is, how do genetic differences make some people more or less susceptible? Okay, so very briefly, I'm going to tell you this story. It's going to take just a few minutes, really. The story begins with the search for a mouse model for COVID-19, which I realized back in March of 2019 was going to be a real roadblock for the field. So we started searching at JAX for the perfect model, and what we came up with was an imperfect model, but has been extraordinarily popular. It existed already. It was something that Paul McRae made in Stan Perlman's lab to study SARS when that outbreak occurred. And in this case, because the SARS viruses predominantly enter the cells through an interaction with the ACE2 receptor, the humanization of that receptor through a transgenic approach offered a way to study SARS, and it turns out SARS-CoV-2 as well. Terrific. The only problem is that it's very, very rapid. The disease progresses, the animals die within a week. Terrific for screening vaccines, not so terrific for looking at the larger, longer problems associated with the disease. And indeed, here's some data from one of the early papers from our collaborators at NIAID showing how these mice, when infected, even with fairly low PFUs of the virus, tend to die quite quickly after weight loss. And so this had, my idea was here, if we could somehow modify the genetic background of this animal very rapidly so as to supply the community with better models, we might be able to better model the actual disease in humans. So very, very simply, we took those diverse mice I told you about, the founders of the collaborative cross, and crossed them with the K18 ACE2 model from Stan's lab. Now, this means that you get the transgene into the mix so that the animals will be infected, but it also dilutes out the diversity in those other mice because you have 50% C57 black 6 background, which was what that gray mouse was on. It was on a C57 black 6J background. So the concept was 50% of the animal will be different, 50% of the animal's genome will be the same. Will it make any difference at all? Fortunately, I had some people who were as crazy as I was to try this. My colleagues at the Rocky Mountain Laboratories, the NIAID outpost in Montana, Sonia Best and Shelley Peterson, who have been just fantastic collaborators. And with their help, we've managed to look at in some detail at how these different models respond. I'm not going to show you all the data. It's on bioarchives if you're interested. And I should say that all the mouse models that I am describing today are now available at the Jackson Laboratory so that you can order them up if anything here looks of interest to you and you're working on this disease. As you can see, in the very middle is the original model. You can see how they really don't do very well. And indeed, they die very quickly after within a week of infection. Now look at the heterozygote over on the left. This is a mouse that is half C57 black 6 and half PWK, which is one of the 8 founder strains for the collaborative cross. These animals walk away, literally walk away, asymptomatic, just like asymptomatic patients in the humans. Whereas some of them, and I'm just showing you 3 examples here, some of them, such as this one, which was a mix of black 6 and NOD, which is known to be a good model for autoimmune disease, turned out to be sexually dimorphic. So the females did pretty well, and they're the ones that are susceptible to autoimmune disease, interestingly. So there's all sorts of magic biology in there to delve into, whereas the males were not so good. So I don't expect you to look at this, but it's just simply to tell you that these are 8 out of the 10 strains that we've looked at. And in every case, they had a unique and interesting set of biology associated with them. just a tiny bit more data for you to show that we saw everything from mild, moderate, severe infectious disease in lung pathology. We also saw brain pathology. And we saw all sorts of other interesting side effects, including thrombosis, etc. Bottom line is that we had 8 genetic backgrounds, even though they were all half C57 Black 6, they managed that genetic background managed to have profound influence on the way these animals survived or did not. Now, for those of you in the field, you're probably familiar with Akiko Iwasaki's work, fantastic work out of Yale. This is one of her slides from a recent talk she gave at JAX, where you can see that her overall message is get in and get out. As an immune response, you need rapid inflammatory response and then rapid cessation of that response, which is canonical with a lot of different diseases. And what she found in many patients looking at, for instance, young versus older disease, low viral exposure versus high viral exposure, in general, this is obviously just a cartoon, but over and over again, it's the delay in the response to this virus that causes the trouble, because then the virus gets away with murder and off it goes. So we were interested to know whether any of this was actually true in the mice that we were looking at. And this is, again, a very complicated slide. I'm going to walk you through it very briefly. This is just some cytokine analysis that we did on the bronchial alveolar fluid of these mice. And knowing what we knew from Akiko's work, we managed to sort of make a dendrogram here showing how these various strains were related to each other. And I'll just tell you very briefly, the take-home message is the following. This is a cluster of cytokines and interferons that are rapidly upregulated in the mice that are resistant to the disease. And we grouped these as a few mice together that we found that were exceptionally resistant, like that PWK mouse I showed you. This is at 3 days post-infection. Now, just compare that to what a mouse that is not going to do very well is expressing. And you can see that in this cluster, these are sensitive strains. There is a very sluggish response. Now, by day 6, as you see over on the extreme right there, everybody's responding, but by that time, it's too late for the sensitive strains. They're going to be in trouble. And we used actually type 1 interferon as a kind of a hallmark of this. And you can see in the next slide, which is a sort of a kind of a matrix, a network for you to look at. And again, this is very preliminary data, but you can see how different the relationship, either positive or negative, is between various immunologically important molecules, including the interferons. And interferon alpha turned out to be one of the hallmarks in both humans and mice. If it came up early, you were probably doing okay. You had to get rid of it quick too, though. So you can see that by 6 days on the right, there's a whole different kind of profile in those animals that were sensitive versus those animals that were resistant. This is just to try to get a point across. It's not obviously fully worked out yet. We just got the data recently. But the most important thing is that with those animals that could actually invoke this rapid response, the clearance of the virus out of the system was rapid and very, very dramatic. So just for instance, compare C57 Black 6, the original strain on the left, where you see that the level of PFUs in the lung is high, high, high throughout 3 and 6 days in both males and females, whereas in PWK, it's absolutely being squashed by 6 days, even though the original infection was extremely high. So this actually looks very, very similar to the way we're beginning to understand the overall immune landscape in patients. And by definition, we're interested at JAX at making even better models. And of course, that's easy to do. We're now just mating everything together. And we're also attacking the problem of, are we really recapitulating the disease by overexpressing a human gene in the mouse? So what we're hoping for is even better models to hand to the community, because ultimately we want to be able to ask the question whether different models are going to mimic different components of such a long-standing problem as long COVID. And so the way we're attacking this is to make models of disease that are on different genetic backgrounds, but including different components of the human response to this virus by introducing different knock-ins and mutations. And obviously, we also have a lot of experience in humanizing the entire immune system. So this is the direction of travel. So how does this relate to the overall question we're addressing today, which is the challenge of this translation? I think one of the ways to think about this, and Shai is going to laugh because I'm a big fan of cubes as well. If you think about a dimension in which you are essentially grabbing the entire information about a genotype of an animal, and then you use phenotypic tests to be able to grab as much data as you can on that score and add environment, you begin to be able to build a matrix in which we may be capable of making much better associations and causality between humans and mice. And of course, we can't do this in entirety, but if you can get the outline of these sort of cube-like matrices, we're hoping that we can make better inroads by using the mouse-human interface as a principle here. And I don't have time to show you, but we've done quite a long study on type 2 diabetes and another one on Alzheimer's, which is really bearing this out in some detail. So the concept here is that if you have data that is somewhat sparse in the human on this score, namely, how do you get genotype, phenotype, and environment, or aging, or any of the other aspects that we can't necessarily always control in a human, you may be able to use mouse data, which is much deeper and more complete, and get those vectors sorted out, and then see which of those vectors actually translate back into human, and then build up the human reservoir of information. And of course, I'm basically telling the same story as Shai, just with different words here. The thing about the mouse that I'd like to point out is that you can start with mice that are diverse, but you can also get cells out of them that are diverse. And that means you can start doing in vitro assays and drug development using the diverse data in the mouse and then translating into human. So I think that this panoply of modeling that we can do, whether it's cellular, physiological, or computational, is really going to be important in grabbing every aspect of the diversity in our own genomes that produce all the human variation that we see. And so our goal, of course, is to make mice that match people as best we can. I'd just like to mention very briefly before I close that the rapid pivoting to work on COVID-19, about which none of these people knew anything when we started, with the help of some fantastic collaborators, both at the NIAID and at Imperial College, where I also have a position, has been absolutely critical to testing out this model and for progressing it further. And I'd like to just, of course, thank the diverse mice, of which we have 11,000. And once more, any of those mice that you see there that you are interested in, please get in touch with me for more data, or just look on our website. And thank you very much for your attention. This is actually what it looks like right now up here. It's daylight, but this is the color of our fall. So do come and visit us as soon as we can lift the restrictions and get us together again. Thank you.
Thank you so much, Nadia. That was a wonderful talk. And I'm curious, you touched on so much in your conversation. One of the things that comes to mind is perhaps the lens of aging plays into this. Obviously, that's another just arm on the matrix that you noted, and where you see that perhaps weaving into this approach.
We have a very, very avid interest in aging and making models of aging that will help us understand better how humans age and what we can do to maintain our health for as long as possible. We are part of a shock center. We also have a large intervention testing program and a center for aging research. What that does is provide us in-house with incredible capacity to look at animals as they age. It's a bugaboo because do you wait 2 years? What are you going to do? You got a postdoc. What's the postdoc going to do in the meantime? So we are essentially trying to figure out ways in which to produce enough mouse models of aging that we can provide the community over and above what the National Institute of Aging can provide with models that are already old. And needless to say, we've started with the canonical inbred models, but we're now considering aging out these other models that we're producing so that we can offer, for instance, collaborative cross cohorts at any age to the community. It's something that we do, in our sleep, is grow mice. So the question is, would these aged mice be of use? I don't think there's any question they would. That said, I have a personal soapbox about if you age a mouse and they're dead bored and they're sitting in a cage for 2 years and at the end they die, are they just dying of boredom? So or if you age the mice and you feed them, high fat, high sugar, or some kind of rapamycin, that's great. But the question is, how does that relate to humans? And so we're actually thinking very hard about the environmental aspects of aging and how we may be able to study phenotype much more effectively if we provide mice with a spectrum of different influences. And that includes diet, it includes all sorts of supplements. It also includes lifestyle, because mice actually have a life of their own, and that's food for another lecture.
Well, I know that we're going to dive deeper into some of these questions during the panel discussion, so I may just park it there. And thank you again for that wonderful presentation, and alert the all members who are watching that we are at break time. So please feel free to continue adding questions to the Q&A if any are outstanding. And we'll be taking a break for about 10 minutes before our next session. And we'll see you back at about 5 past the hour. So thank you so much, and stay tuned. More to come. Hello, everyone, and welcome back to the second half of our session. It's wonderful to start with our third and final speaker of the day, Douglas Brubaker, an assistant professor in the Weldon School of Biomedical Engineering and core faculty in the Regenstrief Center for Healthcare Engineering at Purdue University. His lab develops systems biology approaches to understand mechanisms of host microbiome interactions and approaches to translate biological observations from models to humans. Dr. Brubaker's presentation is titled Computational Humanization of Latent Variables for Cross-Species Modeling. And it's my pleasure to welcome Doug. Take it away.
All right, can everyone hear me? All right, thank you for that introduction. And I really want to thank Alan Frontiers for the invitation to speak today. I started my lab here at Purdue in August 2020, and because of that, much of this talk is going to be retrospective and draw upon work from my postdoc. So I've tried to weave in some thoughts throughout on principles that we can apply going forward to better translate biology from mouse models to humans. Okay. So as I said, I began working on these questions in my postdoc, and that was in Doug Laufenberger's lab at MIT. And of course, we weren't the first folks to start thinking about these challenges of preclinical to clinical translation. But what I think distinguishes recent efforts in this space, and I would put Nadia and Shai's efforts in this area too, is that we're thinking about translation as a methodological problem in and of itself that has its own considerations. And so the thinking developed in the Laufenberger lab was that it could be useful to encode data from a mouse experiment in a computational model in some way, whether that's machine learning methods, biological networks, or even mechanistic ODE models, and that one should use what human data is available to computationally humanize the coefficients and weights in those models, and thereby translate the computational model to human physiological contexts. The overall goal of these efforts being to make a prediction that as closely mirrors what one would find if you had hypothetically been able to do the experiment in humans. And one of the things that is inherent in this way of conceptualizing translation as a methodological problem is that there's not going to be a universal translation model that works well for every jump between mouse and humans. And in fact, some models and approaches may work better than others for different kinds of questions. So we initially proposed 4 categories of translation problems motivated by the biological question, the data types and coverage, and the phenotypes of interest. And of course, what discrepancies one had to accept between mouse and humans. But I think there are likely many more categories than this, and I think it would be helpful for folks to think about those going forward. So our initial efforts in the cross-species modeling space took a cross-species pairs approach, much like what Shai described in his talk. And in our paper, we benchmarked different machine learning approaches for predicting human disease-associated genes and pathways. So let me see if there we go. So we pulled data from 2 papers looking at inflammatory disease gene expression data in mouse and humans, and taking the way that the authors lined up the cross-species pairs. Oh my.
Hold on, Doug, I'll get the slides back to the beginning.
Okay, I think we're okay actually. So taking the gene expression data from these papers and taking the cross-species pairs as the authors defined them, we benchmarked either supervised models that used no human data or semi-supervised approaches that incorporated human data in an unsupervised manner. The goal being to predict disease-associated genes and pathways. So our ground truth here was the human gene to phenotype associations, and our baseline control was what you would get if you took the mouse data as is and compared that to the human. So what we found as we began to aggregate all of the results from the different case studies together and from the different computational approaches, supervised and semi-supervised variants of k-nearest neighbors, support vector machines, a random forest, and a neural network approach, was that you could find general principles of translation. So what we first noted was that any kind of computational modeling seemed to improve upon generalizing the mouse observations on average. And the most efficacious model we found across case studies seemed to be a semi-supervised neural network. And so as we dug into the individual case studies, some general trends that emerged were that pathways translated better than individual genes. We saw that even if the mouse didn't recapitulate any of the human biology, as in some of these sepsis models here, you could still make a pretty good guess about the disease genes and pathways in the human through integration with human data. And then something that I think really echoes what Nadia showed was that if we incorporated multiple genetic strains of mice together to train the model, that increased heterogeneity seemed to improve our predictive power. And so if we abstract a little bit from this study, I think it supports the idea that heterogeneity can be an advantageous feature in cross-species modeling, that this greater genetic diversity from multiple mouse strains could actually better recapitulate human variability. And then that pathways and networks seem to be more translatable than single genes. And that makes sense because these higher level mechanisms are probably more evolutionarily conserved and not subject to evolutionary pressure in the same way that individual genes are. But then this begs a question going forward. If we're going to focus on higher level biological mechanisms, how should we encode these multivariate relationships? And if we want to bring in data other than gene expression data, how do we incorporate multi-omic parameters into those models? Well, for this, I'm going to shift gears and talk about another study in which we sought to translate mouse proteomic information to a human gene expression data set with the goal of understanding drug resistance in inflammatory bowel disease patients. The clinical problem that we were trying to address here was that Crohn's and colitis patients progress through a fairly predictable treatment regimen that almost always ends with treatment with an anti-TNF-alpha biologic therapy. Now, of those patients that receive anti-TNF agents, about 40% will show primary resistance, and 2/3 of those that initially respond will show secondary resistance. Now, the way in which folks have mostly thought about addressing this challenge has been to look at bulk gene expression measurements from intestinal pinch biopsies. And then taking that data, they try to identify disease-associated genes and then gene signatures pre-treatment that can predict responsiveness to anti-TNF therapy. But as many in this audience probably are aware, RNA is not always a good proxy for protein function, and it is possible that important targets are being overlooked. So to fill that gap, we had access to 2 high-coverage IBD mouse model proteomics data sets, in which we had previously studied inflammatory signaling networks in a very controlled experimental context. And so the challenge we sought to address was to translate the mouse proteomic information to humans. But besides this jump in species, we also were trying to jump from a mass spec-based proteomics experiment to a microarray-based transcriptomics context. And then I think more critically, there was this phenotypic discrepancy we had to consider. These mouse models were never treated with an anti-TNF agent. And so what we were wanting to do was relate a control versus inflamed contrast in mice to a therapeutic contrast, anti-TNF responsive or resistant in humans. So the method that we developed to try to address these challenges is termed translatable components regression. I think the slides are behind again. And this was published last year in Science Signaling, which conceptually is a projection of human samples into a mouse latent variable space. And so our approach here was to first take the mouse proteomics data and construct a PCA model, which encoded in lower dimensional vectors the most important proteomic variation between mice. And then we projected the human RNA samples into this mouse proteomic coordinate system. What this gave us was a representation of the relative differences between human samples in terms of the mouse coordinate system. From there, we could build a regression model to relate the human and mouse principal component projections to the human therapeutic response phenotypes. This allowed us to identify mouse principal components, i.e. proteomic networks, predictive of human drug response phenotypes. And the thing about this model is that it allowed us to look at not just those high rank principal components that strongly explain variability between mice, but to consider those lower rank principal components that maybe would be overlooked otherwise. So I'm showing you here on the next slide what this looks like in practice. So if we project human Crohn's patients into a TNF-ARE mouse principal component space, we see that mouse principal components 1 and 6 are capable of separating responsive patients from non-responsive patients. And in fact, these were the two most predictive components as identified by our regression model. And if you look at the proteins that are driving separation along these axes, on mouse principal component 1, it was an immune cell trafficking integrin network for which drugs already exist, but these are not remission-inducing therapies. And then on mouse principal component 6, which only explains about 4% variability between mice, the proteins that were strongly weighted on that were collagen-binding integrins. And so this was the network that we decided to follow up on experimentally. And in the next slide, we designed a couple of follow-up experiments to look at this network in more detail. And so what we did was, with collaborator Ken Lau at Vanderbilt University, looked at single-cell RNA sequencing data from a post-infliximab-treated Crohn's patient, so colonic Crohn's disease, an anti-TNF agent. And we were able to identify the cell subsets that expressed the genes in our mouse principal component network. These seem to implicate an activated T cell subset. And so what we then did was designed a follow-up experiment using human PBMCs, in which we could inhibit collagen-binding integrin subunit integrin alpha-1, we could inhibit TNF, and then inhibit the combination. And what we saw was that there was greater overall cytokine suppression in the combination treatment than with anti-TNF agents alone. And so in the next slide, I think one of the takeaways from this study, and you can find it in Science Signaling to read more details, is that we were able to pull out a potentially novel drug resistance mechanism for Crohn's disease patients that was encoded on a low-rank mouse principal component. And so this adds an interesting principle to our list for cross-species modeling, that it's potentially these lower signal mouse biological networks that could be consequential and translatable to humans. But this also raises an issue of how we know when to care about these low signal networks and why this would work in the first place. So to answer that question on the next slide, I want to briefly touch on some exciting work led by a graduate student at the Laufenberger Lab, Mi-Lim Lee, whom I worked at MIT and have continued to work with since moving to Purdue. And in this paper that just came out in Frontiers in Neuroscience, Mi-Lim applies the Transcomp-R framework to look at the translatability of different Alzheimer's disease mouse models. There were a number of really interesting innovations in this paper, but I just want to highlight a couple here. One being that she flips the model, and instead of projecting human into mouse, she projects mouse samples into a human principal component space. The idea here being that if the human principal components separate the mouse disease versus control, then the disease contrast in the mouse is well represented in the human-to-human variability. So on the next slide, the other two things I want to highlight from this paper are that we devised a way to calculate the variance explained in a mouse by human principal components. And this explains maybe a little bit of why Transcomp-R is picking up on those low-rank mouse PCs in the Crohn's disease example. So in the bar plot here, looking at the green bars, these are variance explained in human data by human principal components, and it's in descending order of importance like we would expect. Now, if you then calculate the percent variance explained in a mouse data set, the variance explained is all over the place. And we can actually find human principal components that better explain mouse variability than they do human variability. Another thing that Mi-Lim did that was really interesting was she developed a post hoc test to correct for the age of the mouse in assessing whether or not we could predict disease status. So what I'm showing here is the effect of that correction on the human principal components for predicting mouse AD status versus control. And just to give you an example, principal component 9 here, more important in the mouse than it is in a human, and it has a durable disease signature. Whereas principal component 14, even though it's more highly predictive in the mouse than the human, that signature is entirely confounded by age. So moving on to the next slide, something that we have done in my own lab, building upon some of the ideas proposed in this work with Milim, is to apply transcomp-R in the context of post-traumatic osteoarthritis mouse models. So we've moved from pan-inflammatory modeling to Crohn's disease, to Alzheimer's, and now to a musculoskeletal pathology. And in the osteoarthritis field, there are many different mouse models one can use. Surgical induction with ACL tears or with destabilizing the medial meniscus, or chemically induced mouse models. And these all have different structural and inflammatory consequences. And this work was done in collaboration with another new BME faculty member, Dr. Diva Chan, who moved her lab here from Rensselaer Polytechnic at the same time that I came here to start my lab. And so this is truly a collaboration born out of 2 pandemic startup labs. And so in the next slide, I'll show you some of the key results from this collaboration, and we're working on a preprint to highlight these results. But I think the major takeaway I want to leave you with today is that in cross-species modeling, considering the human clinical and demographic covariates can crucially influence how we interpret how translatable the mouse biology is going to be. So this is work done by graduate student Megan Pendiala and undergraduate student Stephen Douglas, fantastic students to work with. And what they show is that by conventional differential expression analysis, we wouldn't expect very high correspondence between either the DMM or ACL tear mouse models of osteoarthritis. But then building a transcomp-R model on that same data, we can see a couple of interesting things happen as we consider the predictive power of the mouse principal components on their own, and then what happens if we condition them on human age or human sex as a biological variable, both of which being known risk factors for osteoarthritis. So in some cases, conditioning on human clinical variables can improve the translatability of the mouse model and highlight biology that we really want to care about. And in some cases, it can completely explain away the translatability of the mouse model. But one of the things we were able to pull out that was interesting and surprising was that we could condition data from an all-male mouse cohort on human sex as a biological variable and find a stronger signal. And what this tells us is that there's a sexually dimorphic gene expression effect that we can detect from these mouse samples that could be relevant in humans that wasn't apparent right away because the original mouse experiment wasn't balanced for sex. And so to kind of add this to our list of translation principles on the next slide, and to kind of leave you with what I think is a really exciting future direction in this field. I think there's tremendous opportunity for us to bring clinical and demographic variables into cross-species modeling, both to increase the effectiveness of translating biology from mouse to human, but also to increase the equity in the drug development process by including age, sex, and ethnicity as variables we consider when looking for more effective treatments for complex disease. So in the last slide here, I just want to thank all members of my lab here at Purdue, our collaborators, especially the Chan Lab and those in the Laufenberger Lab, and then to acknowledge the generous support that we receive from Open Philanthropy to further our work in cross-species modeling of Alzheimer's disease, and then support we receive from the NSF as part of this Biological Integration Institute just launched at Purdue University. And we're recruiting grad students and postdocs for all of these projects. And thank you so much.
Thank you so much, Doug. That was a wonderful talk. I know that we just have a little bit of time here, so I'm curious, when you're doing any of this work, obviously it so depends on the quality and quantity and breadth of data that you have available. So how much is that a challenge for making these models and for looking at all these aspects?
I think in that question of how much data you use, there's going to be a balance between specificity and generality. So if we have large data sets for a specific context, then we can reduce the variability in the model and have a better prediction going from mouse to human. If we incorporate lots of data from different contexts, then we might have a highly generalizable method that doesn't tune specifically well to particular disease areas. So I think there's a balance there between.
I know you may have noticed one of the previous questions asked of Shai was how does your algorithm or how does your approach know when it could be buttressed by a specific flavor or type of data, or if there's missing data? And I guess it's how do you know when you don't have something? and I wasn't sure. There's a couple different ways Shai answered it, but I guess I'm going to put you on the spot of how much of a challenge is that as well?
So how do we know what we don't know in cross-species modeling?
Yeah.
So I think one of the things that I really like about the transcomp-R approach and using these sort of inferential models is that we can get a quantitative measure in terms of classification error or in terms of the regression p-value or other metrics of how well we're doing at predicting a specific outcome. And if we can't do well with the data we have, then that's maybe a suggestion that we need to look for different kinds of molecular data because it could be a different level of regulation that's important. Or perhaps we're not conditioning on the appropriate clinical or demographic variables. So essentially model failure is an indication we need to look harder and look for more data.
Being at the bleeding edge, it's always exciting to see how one takes these approaches and where to go next. And so I see that we are now right on our time for our discussion panel. So I will transition quickly to that. Thank you again, Doug. And I'm going to ask all of our three speakers and our moderator to join us back on camera. And it's my pleasure to introduce our moderator for today's panel discussion, Dr. Stacey Finley. She is the Gordon S. Marshall Early Career Chair and Associate Professor of Biomedical Engineering at the University of Southern California, and the founder and director of the Center for Computational Modeling of Cancer at USC. Dr. Finley's research group uses multi-scale computational models to answer unresolved questions about behavior and to identify new ways to control cell behavior. She's applied these models to study signaling that promotes angiogenesis, metabolic pathways that cells use to proliferate, and immune cell activation. So thank you for joining me, Stacey, Doug, Nadia, and Shai. Stacey, I'll pass it to you for the opening question.
Sure. Well, first, let me thank you, Dr. Richmond, and the Frontiers group for this phenomenal panel. Just a great session. All three talks were excellent. I really enjoyed listening to them. And so what I get to do, I have the great pleasure and honor to be able to talk with the three speakers and Dr. Richmond as well to think about how to synthesize some of the ideas and concepts that came up, and then also how to be a little bit more forward thinking about what comes next. So maybe I'll start with one question, which is, can you maybe highlight or emphasize some areas or examples where it has been very successful making that translation from mouse models to human physiology? In all three of the talks, I think there was a lot of exciting data and great examples. Can we maybe highlight one or two that are really exemplary and that we can maybe glean some insights from in terms of moving forward too? I'll throw it out to Shai. Why don't you get us started?
Okay. So I think it's a wonderful question, and I think in many ways my answer is has two parts. So we all shown I think initial conditions where you can see this works, right? But but really this is a field in its infancy, and I think that there lies the great potential. And I kind of try to hint at this at the beginning of my talk. I think it's only now that we're coming to realize on the one hand the traditional way we've been using mice It has caveats. And the reason we realize this is that suddenly there's data to compare against, and that we realize we can overcome this. And it doesn't matter if we're doing it from using the genetic diversity or changing to wildlife, kind of wild strains, or computational models. The data is there to bridge that gap. And so we if you look at our, for instance, the results I showed, success, basically we're to the point where we're shifting the predictions by on average 30%. That's a huge win for researchers. On the other hand, it's just gene expression in a bunch of different diseases that we can, mostly inflammatory diseases. There's so much, Doug showed there's multiple different ways where you can kind of enhance this more. I think it's early days, and I think the community of people who can use this, which are people who traditionally have been using mouse models, this is very foreign for them yet. And so there's even a bridging of a gap that comes from bringing these kind of new thinking about mouse biology and how to use data and genetic diverse strains, which also have some, they're not cheap to do, to the researchers, who this is how they've been doing their basic research, their entire professional career. So long road ahead, very excited.
Nadia or Doug, do you have others to add? Examples that you're really excited about that maybe we can glean more information from in moving forward?
I could start just maybe keeping to a relevant topic given what Doug just described. We do have a fairly active Alzheimer's research program at the JACS. And I'd say I'm not an Alzheimer's expert, but what I've taken away from the data so far that has been generated using some of our more advanced mouse diversity panels and looking at how to translate to human in some of the same ways that Doug described, where you start with the mouse and ask, do the humans match the mouse? We've discovered that with enough genetic diversity in the mouse population, you can pick up the subtypes of Alzheimer's disease that you see in the human and find correlates in the mouse that can be much better defined because we have access to all sorts of tissue data. We also, of course, can start pinpointing genetic loci that are important, whether they be non-coding or coding. So for instance, there's a inflammatory component to Alzheimer's that really distinguishes certain kinds of patients from, let's say, a neurological, and those subsets of patients can actually be mimicked in subsets of mice, so that then, of course, you can dig deeper into the mechanism in the mouse to inform better how to predict the subtype of dementia that might be addressed with drugs. So it's just one example. I think we could all give examples of this. And the most important thing is it's really early days. And I can't stress enough my agreement and concordance with what Shai is saying about tradition, habit, scientific bias, a sense of, well, this is the way we've always done it. We're going to need all that AI to get down to actually seeing the patterns that are now becoming evident. But we're also going to need huge amounts of phenotypic data to be able to really unravel what's going on in GWAS and human. Transcription isn't enough. I mean, we're going to be looking at every level. And I think that that's where the proteomic approach is so important. So I'll leave Doug to talk about that.
I think to go to the original question real quick about approaches we're excited about, I hadn't seen the SARS-CoV-2 collaborative cross models, and I thought those were really exciting because it sort of gets at this one side, make the mouse model more diverse, representative of human. But then I think where we also need to do some work is in what human cohorts we do molecular profiling on. Most of what's in the literature is men, it's mostly white men. And so those data sets, when we're doing this kind of translational modeling, don't give us a lot of diversity to work with in terms of the modeling. So whether it's more diverse cohorts for transcriptional profiling or proteomic profiling, which gives us, I think, a better target to shoot for in humans if we can. I think those are really exciting approaches, and I think we could do more of going forward.
Yeah, so something that Oh, go ahead, please, Shai.
I was just gonna say, if I didn't emphasize it in my talk, but some of the analysis, having built this database, some of the things we did was actually also compare within humans, right, and within species, and just to kind of, so the numbers there are still horrendous, right, in terms of our ability to reproduce, or not horrendous as much as they tell you how much heterogeneity exists in biology. You saw this in some of my comparisons. So certainly, moving to kind of leveraging this diversity, not as an average, but as Doug said, leveraging the heterogeneity to actually build better models is the way forward to do this. And along the same lines, and just kind of looking for success, we recently did with collaborators here at the Technion. We basically took a human anti-cancer immune checkpoint inhibitor drug and applied it and generated a diversity of mouse models which are non-responding. And just that diversity cleans out a huge amount of the things that are just appear in one of the mice, as Nadia showed, and we're able to identify a successful human validated biomarker for response that works better than anything that's currently in this case in melanoma. So that it's just recognizing that you need to embrace the diversity and not kind of shy from it, which is how mouse researchers for many years kind of treated their perfect experimental system. So.
Yeah, I think this gets to a point that a direction I wanted to move into as well. Thinking about what Nadia said in incorporating information about aging, about the response, about the environment, and also thinking about what Doug said in terms of not focusing on a particular gene, but actually looking at the pathway and the systems level. How has this systems perspective and the role of that and the importance of that evolved when we think about translating information from mouse to human? That systems perspective is hugely important. So how has it evolved in this field?
I guess I'll start by answering. I think there's been a much greater appreciation that there needs to be a dialogue between sort of the snapshot disease control, omic profiling, but then dynamic experiments, watching how these things change over time. And that we need to, the only way that you can deal with these multiple levels of regulation, these multiple interacting molecules, is by using these mathematical approaches that embrace the complexity of the system. Whenever we try to isolate single genes, or even to some extent single pathways, we can always find things that don't match up between species. But I think at the systems level, because there is this unity in biology from the evolutionary perspective, that embracing complexity actually gets us closer to a more predictive way to translate across contexts.
Maybe just to add on Doug's answer, I think we all recognize that the network is the thing that we need to model in the end. I also think that if you think even about the traditional way, you knocked out a particular gene that looked awesome, that had a good phenotype, you were happy, but as soon as you start interacting, you lose it. And so we know we need to recognize, look at the network. There's a problem in this field, and this has nothing to do with cross species, which we don't do a great job yet in entirety in modeling the networks accurately. And if you look at people who have kind of been trying to benchmark network inference and so forth, it's not like this is a mature field yet. And so doing the transition across species is even a larger problem. I think much of what Doug showed is some way around it is getting the network effects through dimensionality reduction, rather than trying to model the network itself, which we're not there yet. And there's some early work where people have tried doing this using single cell. I entered in the Q&A on single cell data from Fabian Tissa's group, and basically what they're trying to get, with these kind of more kind of bigger network approaches, is you get the top genes, the ones that you probably would have picked up, mostly anyhow. So we have a ways to go, and fundamentally we need to fix the network inference problem.
Mm.
Just from a mouse perspective, if you think about all the differences between mice and human, including things like how they've been environmentally sort of shaped evolutionarily, what they eat, how they live, what are the real drivers of their evolution, it's going to be different. Obviously, it's going to be way different. So then the question is, how do you suss out the commonality, which is going to be in the genes because we know how similar we are. So there are going to be sort of, I think, there's going to be noise in the system that we have to embrace, and we have to kind of identify it. I'm just saying what Doug said much more eloquently. And I just feel that, trying to make mice into tiny little humans isn't the point. And so therefore, humanizing this gene and humanizing that gene, it's a bit short-sighted in my view, because we really do need to think in terms of the whole organism and how that organism has evolved. And when we understand that better, we can pull out the things that are important that we can use as models, and the other things we're going to have to leave behind.
If I may, to follow up on what Nadia said, I think, and the other way of looking at this problem is not that we're going to get great at doing this from a, we'll model the network, but rather we'll get much more pinpointed at what is the utility of anything you're doing. And we saw this in some of this in Doug's talk, right? I mentioned it as well. It's not about, can I get the perfect mouse model? Is if I need to do a mouse model for a toxicity study or an efficacy or understand what's happening in tissue, I kind of hinted at this early in my talk, then I can build a machine learning model that does this very specific thing, and I've geared towards it, and eventually I will get it much better. I'm not, it's much more difficult to do a generic machine model that will move us from the mouse to the human in all aspects. That's going to work well.
Yeah. No, I mean, it goes back to the quote from George Box that you put in your talk that we all embrace, that not all models are correct, right? But some are useful. So it's how can we begin to educate ourselves and the field as well about what are the mouse models uniquely and well suited to address, and then where are they, woefully inadequate. So how can we start to separate out where mouse models are a very good place to start, and then other places where it's not the right tool. Just like what we do with our computational models, right? Black box versus mechanistic, ODE versus agent-based models. Like, how can we better classify the place, the role where mouse models can really highlight and be emphasized?
Well, from my perspective, I just want to echo something that Shai just said about the perfect mouse model, and specifically about how there may be deficiencies in the actual human data, which we also heard about from both my colleagues. And so before you jump into a mouse model that is phenotypically appropriate, let's say, because it mimics what we see in a disease, I think we have to ask, what did we model in the human disease first? Because often that is a very, very tortuous road, and there could be a lot of false positives in there. So I just want to point out that better phenotyping of humans is going to be critical to this whole enterprise. And of course, I'm championing better phenotyping of mice to go along with that, because then hopefully we'll be able to meet somewhere in the middle. And I do think that a lot of the phenotyping that we do in mice can be quite biased by environmental stimuli, by genetic background, by all sorts of other issues that we could go into. And certainly, I think the clinicians agree that electronic health records are a complicated beast, and that it's not going to be a one-to-one correlation between what we see in the human and the mouse, maybe because we just don't have the data that will allow us to make that obvious correlation. So I'm just, it's caveat emptor when you ask someone at the Jackson Laboratory, I have a disease. What's the mouse model I should buy? And it's, I'm pulling my hair out. Yeah, how long is a piece of string? It's going to be very difficult because we are not embracing, right back to what Shai said at the very outset of this seminar series, which is we are not necessarily setting up ourselves for success because we're not asking the right question about disease or a set of patients or even a way of measuring health. I think so. These are issues that get way beyond just how do we pick the right mouse model and gets really into human biology, which I think is where we're heading anyway.
And I think this idea of better phenotyping the mouse, better coverage of phenotypes, and going one step further into the range of perturbations we can do, that space will always be greater for what we can do in a mouse model than in humans, because there are just things we would never try in person as a first pass. And so if you want to see what something does to a living organism and be able to do that at scale with any kind of statistical power, the mouse is perfect for that. But then I think when we want to move into humans, there's this intermediate role for sort of humanized microphysiological systems, where we can then perturb the human cellular network based on what we see is efficacious in a live organism. And so it's this sort of march of science from preclinical to clinical work, I think.
Yeah, that definitely goes back to something that Nadia sort of ended with, in thinking about how to advance diversity and thinking about the cellular scale, the physiological scale, the computational modeling that can help that as well. So what are some additional either experimental models, computational tools that can help bridge this gap? We can think about, right, what we would like to do, but what's the availability of the toolkit that we have at hand?
I think that's a wonderful question, and the availability is enormous because it's such a young field, right? So first of all, start with the single cell data that is now easily, just tons of it are coming out all the time, and is perfectly fit to augment these types of approaches. It ain't perfect data, there is no such thing, but there's a huge amount of it, and so that is eventually, once we learn how to use it for this problem, it's going to be advantageous. But we also can think about this once you start thinking about this from a computational perspective. You don't think about this, I think, anymore. You shouldn't be thinking about this as a single leap from mouse to man. There's a lot of species in the middle that you can start modeling computationally, right? And start inferring that and narrowing, your confidence interval, on particular behaviors. So I think those, at least to me, are the two most exciting places. Doug mentioned multiple different data types. I mean, there's just endless new things that can be done. Just to kind of address something that you said before in terms of the use, I said this in my talk, but I want to emphasize this. So I've been talking a lot with pharma companies, and what I'm hearing basically is that the idea in pharma, literally, they said the discovery in the mouse is gone. It's no longer something that people do in pharma. Now, obviously, this is could be a slightly stronger statement than is true, but I see this with the type of stuff we do, which is let's bring all the human data to the earliest stage. Now, what it does is two things. The big thing is you're making discoveries in human, but it really, really targets what you're going to do in the mouse experiment. And that, from a perspective of successful translation, is a very big deal, because you're not, again, you're doing predictions on a small thing is much easier than trying to do the whole phenotype.
Yeah, maybe.
Oh, sorry.
Go ahead.
I was just going to say that I think this idea of bringing genetics into cell biology is something where the mouse can really help, because you can discover things with genetics that are hard to discover otherwise, because it's an unbiased approach, and rather than single gene engineering or even sort of directed mutagenesis, the the approach that I think is getting a lot of press right now, at least in the aging field, is how do cells actually get reprogrammed back to some useful state? And the lack of really any interest in how genetics might control that is something that I think we need to fix, because it's going to be hard to do this in a human context because we're so diverse and so variable, and because these cells come from people who have lived incredibly different lives and have had incredibly different backgrounds, genetically and otherwise. But in the mouse, you can actually control a lot of those variables, and then you can ask questions about whether a particular phenotype in a dish, which is where pharma needs to end up anyway, for discovery, it's the way they normally go, is something that can be dissected at a genetic level, which then allows you to get into systems biology and all the rest of it. So I'm a big proponent of keeping the cell biologists aware of where their cell is coming from and where the genetics of that original, their original organism, be it a mouse or a human, actually plays out in phenotypes in the in vivo situation as well.
Well, so we're getting close to the end of the hour. I wanted to pose one last question to each of you, especially thinking about sort of where we go next. And it came up multiple times that this is a nascent field. It's evolving, it's growing. And so that means that we have a unique opportunity to start to shape the field in the direction that we want it to move, rather than like going along for the ride. So if you could, think about how to be intentional and strategic about how the field in terms of translating biology from mouse to human would evolve, what would you really be excited about doing, about seeing in the next 10 years?
Can I start?
Yes, please.
So the thing I would most be excited about, I think from the closest thing, right, that you can start tomorrow, is meshing the type of approaches that Doug and I showed with the genetic diversity models that Nadia has been playing with, because then you really leverage the heterogeneity and cleaning it up. I think there's going to be tremendous potential there. Other species and other data types, which are going to fill in missing blanks. And maybe the long-term 10 years that I envision here, that I think is really is not from a scientific side as much as this is an extremely important problem, very broad for biomedical research, that has potential for putting large consortia of academia and industry for it, because it's a pre-competitive problem, right? And it's broad, the entirety of biomedical research. And so I think there's just a lot of potential here to grow and bring in the right data from there's a lot of interested parties that can really change where we are at this problem, which is just getting going, right? I mean, our papers here on our works on this are basically, a few years old. And so I'm excited by that potential, because it's a huge community that's going to grow.
I echo everything Shai said. Just from my perspective, sitting here in the middle of a mouse sort of emporium, I would say that something that was mentioned in one of the talks is, this isn't a cheap enterprise using experimental animals. And I agree with that. And it's also really problematic if somebody wants to jump into this field from, let's say, a computational space like yours, or from a clinical standpoint. People who come across really interesting questions in their patients and then want to get into a mouse project. How do they actually do that? There's so much infrastructure you need in order to really do a good job at modeling a disease. It's not as easy as dialing up a JAX mouse. And so one of the things we're trying to do at JAX is think in terms of how can we enable the community to take advantage of everything we've built at the JAX and other places that have large scale mouse experimentation capacity, to really enrich what's going on in these more computational fields, to help the human GWAS community function on their variation, and to actually bring this genetic variation idea into people's labs via cells versus mice, because it's cheaper to do it that way. So that's what I'm excited about, trying to make that happen over the next 5 to 10 years, is to really enable the community to have access to these really powerful tools, both in model organisms, but also in the data that we derive from them.
I guess in terms of future directions, I'm excited about something that none of us really talked about today were dynamics and modeling temporal changes and responses to interventions over time. I think that's going to be a really hard problem that folks should think about. I think there's a really interesting opportunity to develop computational tools to better tailor the mouse to human translation efforts to particular populations, particularly where data might be sparse or there might be an unmet clinical need. But I think big picture, we really need to think more broadly about the full taxonomy of translation problems that are out there, and what to figure out what we don't know how to do well, and then to focus efforts in those areas, because I think we're going to need to do a lot more work.
Yeah. Well, thanks so much. I have enjoyed, I think it's been a great pleasure for me to sort of pick your brain and to encourage others to dig into some of those outstanding areas and questions that we really are seeing emerging from this field and from these approaches. So I really appreciate your time, and I'll let Dr. Richmond help us wrap up.
Excellent. And I can only echo what's already been said. Wonderful discussion, wonderful topic that you all are being focused on, and you've all had great insights on today. Thank you, Doug, Nadia, Shai, and Stacey. Really looking forward to seeing how this goes forward and how the community can leverage this and how it can grow. I also want to give a sincere thanks to all the folks who have helped make this event possible. The Frontiers Group team, Alexander Bassford, Casey Elkins, and Nicole Huber, our amazing communications team, and of course, our founder, Paul Allen. And to the audience, thank you for your enthusiasm and questions. I see there are a couple of questions that are sneaking in. If our speakers have time, if they want to be able to look at those before they depart. And thank you all for joining us for this Exploring Frontiers session. And we look forward to hosting you at future Frontier Groups event. And please feel free to visit our website to learn more about our upcoming events and our activities. But wonderful session. And again, so much excitement on this topic. So thank you all.
Thank you.